Hello students, welcome once again to the channel Physiology Redefined. In last few videos, I discussed the action potential, different aspects of the action potential, along with the action potential that is seen in skeletal muscle, neuronal tissues or nerve fiber, cardiac muscle, along with the special junctional tissues of the heart. So now is regarding the action potential in smooth muscles. Action potential in smooth muscles. You might know that smooth muscles are the muscles which are mostly present in the hollow visceral tract, hollow visceral organs or hollow visceral tracts. And there are two categories of the smooth muscles, two types of the smooth muscles. What are those types? So, two types of the smooth muscles. What are those types? First one is the single unit smooth muscle. Single unit smooth muscle. So what is this single unit smooth muscle? Single unit smooth muscles are present on the wall of the hollow visceral organs like the gastrointestinal tract, like the blood vessels, all of the blood vessels. So these are present on the wall of hollow visceral organs, hollow visceral organs or tracts. And these cells, these smooth muscles are connected with each other in the particular tissue. That means in the entire GIT, all the smooth muscles are connected with each other by the help of gap junctions. That means these single unit smooth muscles have gap junctions. And as you know that through the gap junctions, action potential or the impulse can very rapidly pass from one cell to others. And thereby all the cells simultaneously stimulated and thereby all the cells simultaneously contract or give the response. That means because of the presence of the gap junctions, all the cells act in a concert and act simultaneously, and this property is known as the syncytial property. So this has got syncytial property. Syncytial property. This has got the syncytial property. And the second category of the smooth muscles are known as the multi-unit smooth muscles. Multi-unit smooth muscles. So multi-unit smooth muscles are present on the wall of respiratory linings or tracts of the respiratory linings, tract of the respiratory linings. On the tract or inner linings, tract of the respiratory lining, and these are having no gap junctions. These multi-units smooth muscles are not having any gap junctions. As they do not have any gap junctions, they function single-handedly. They do not act in concert. So this type of nature is known as the non-syncytial. Non-syncytial. So or no presence of syncytium. So these are the two types of the smooth muscles which are present in our body. But the action potential in these smooth muscles are not same, they vary. That means action potential in the different type of smooth muscles vary differently. So in this video, I will discuss the all type of action potentials that can be seen in all type of smooth muscles one by one. So please keep watching this video till the end to get the clear concept regarding these topics. So first I will talk about the action potential in single unit smooth muscle. Action potential in single unit smooth muscle. Now, single unit smooth muscles, these are present on the wall of the gastrointestinal tract. These are also present on the wall of the blood vessels, and these are also present on the wall of the uterus. So, first I will talk about the action potential that is seen in case of single unit smooth muscle. Single unit smooth muscle, that is the in GIT. So first I will talk about the in gastrointestinal tract. So in the gastrointestinal tract, what's happened? The action potential, the membrane potential is the slow wave membrane potential. So this is the duration in millisecond and the electromotive force in millivolt. The smooth muscles of the gastrointestinal tract, they are having the resting membrane potential minus 50 millivolt. Now this membrane potential that is seen in case of gastrointestinal cells, that is the slow wave membrane potential. Slow wave membrane potential. Slow wave membrane potential. And this slow wave membrane potential constitute the slow wave. This is known as the slow wave pacemaker potential. Slow wave pacemaker potential. And this slow wave pacemaker potential is initiated by a special type of cells present on the wall of the intestine, especially in the wall of the duodenum, that is known as the interstitial cells of Kagel. So these cells of the Kagels are like the S node. That means they are having the capacity to generate the action potential. They have got the capacity to generate the change of the membrane potential. And this slow wave pacemaker potential is one form of the graded potential, one form of the generated potential, which has got the capacity to generate the action potential or the spike potential. But these slow wave potential are not responsible for, they do not cause any contractions. No contraction is initiated, no contraction of the GI muscle. So this wave cannot cause the contractions of the GI tract. And this slow wave potential is because of the opening of the voltage gated calcium channel. So this is because of the opening of voltage gated calcium channels. And as you know, as the calcium channels are the slow channels, there is a slow entry of the calcium ion. As the calcium ion enters slowly, there is a gradual fall of the inside electronegativity towards minus 40 millivolt, followed by gradual closure of the calcium channels. So gradual in stoppage of the entry of the calcium ions, and thereby gradual fall of the electronegativity again towards the minus 50 millivolt. So this is due to the effect of the opening and closure of the voltage gated calcium channel. So slow wave pacemaker potential is basically because of the single channels, that is the voltage gated slow calcium channels. So here you can write down that means the action potential seen in GIT is known as the slow wave pacemaker potential. Slow wave pacemaker potential generated by interstitial cells of Kajal, and this is due to this result the gradual fall followed by gradual rise of potential, gradual fall followed by gradual rise of the potential, gradual fall of the potential difference followed by gradual rise of the potential difference. And this is due to opening of voltage gated calcium channels followed by closure of voltage gated calcium channels, and thereby there is a entry of the calcium ions or exit of the calcium ions, and this results the slow wave pacemaker potential. Now by doing this, this slow wave potential once getting generated continuously, sometime what's happened, this once the slow wave pacemaker potential will reach at the firing level, will reach at the minus 40 millivolt, then what's happened, once this slow wave pacemaker potential will reach at the minus 40 millivolt, that is known as the firing level. So this is the firing level. At this part, what's happened, it induces the spike potential. This induces spike potential. So this induces spike potential. So this one is known as the spike potential. This one is known as the spike potential or action potential. Spike potential or action potential. And this spike potential or action potential, they results the contractions of the muscles or contractions of the GIT. So contractions of the GIT is due to the spike potential that is generated automatically by the pacemaker potential, by the slow wave pacemaker potential, and this spike potential has got depolarization phase and the repolarization phase, a rapid rise of inside electropositivity from -40 to 0, and there is a followed by rapid fall of electropositivity from +0 to -40, and thereby the slow wave potential, slow wave pacemaker potential will take the charge. So this is the spike potential that is seen in case of once the pacemaker potential reaches at a firing level. So what we can see now, once the slow wave potential, pacemaker potential, slow wave pacemaker potential reaches at firing level, then what's happened? There is a initiation or generation of spike potential or action potential that is responsible for contraction of the GIT. This is responsible for contractions of the GIT. Now here question comes, how this action potential or spike potential is getting developed. Now once the firing level is reached, slow wave pacemaker potential reaches at the firing level. At these parts, what's happened? There is a opening of voltage gated calcium channels. Mainly, along with the opening of voltage gated sodium channels. So as calcium channels open, calcium ion enters in. As sodium channel opens, sodium ion also enters in due to concentration gradient. As both the positive ions are coming in very rapidly, there is a rapid fall of the inside electronegativity to reaches at the 0. Thereby, and that one is known as this phase is known as the depolarization. So this is the depolarization of the phase of the spike potential or action potential, which is due to the sodium entry and the calcium entry because of the opening of the voltage gated calcium channels and voltage gated sodium channels. So in this action potential, so this spike potential, is due to what? First one is the depolarization phase. So depolarization phase of the spike potential is due to number 1, opening of voltage gated calcium channels. So calcium entry take place, at the same time opening of voltage gated sodium channels. So sodium entry take place. So both the sodium calcium entry and the sodium entry, these are collectively responsible for rapid fall of electronegativity inside, which is known as the depolarization. Now once the value becomes 0 at this particular moment, this is the phase which is known as the repolarization phase. So this is the phase which is known as the repolarization. So this is the depolarization and this is the repolarization. Upward one depolarization, downward one repolarization. So this repolarization at this particular point, what's happened? There is a closure of calcium channels, there is a closure of sodium channels, plus there is a opening of voltage gated potassium channels. So three things are happening here. Whatever calcium channels and sodium channels are open, they will now close. As a result, no more calcium entry, no more sodium entry. So no further rise of the electropositivity. From 0, it will not go further up. Electropositivity will not increase further. Curve will not go up. On the other hand, due to the opening of the voltage gated potassium channels, and as the potassium concentration is very high inside, potassium ion will go out. Potassium ion will go out. As the potassium ion, positive ions will go out, there is a rapid fall of the inside electronegativity towards minus 40 millivolt, and thereby that is the factor that is responsible for repolarization. So repolarization, so second part of the spike potential is known as the repolarization, and this repolarization is again due to number one. Now here, instead of opening, there is a closure of voltage gated calcium channels, closure of voltage gated sodium channels, and third one is opening of voltage gated potassium channels. As calcium channels close, so no calcium entry. As sodium channels close, so no sodium entry, and as potassium channels now open, there is a potassium exit. Now all these three factors are responsible for rapid rise of electronegativity inside, which is known as the repolarization. So this is how the depolarization and the repolarization of the spike potential will take place. And this is about the action potential that is seen in case of gastrointestinal tract. That means in the smooth muscles of the GIT has got two types of the membrane potential changes. One is the slow wave potential, that is the spontaneous one, which doesn't cause the contractions of the muscle, contractions of the GI tract. And once this slow wave pacemaker potential reaches at the firing level, then only the spike potential will originate, will generate, and that is responsible for contractions of the GIT. So this is how the action potential in case of single unit smooth muscle. One of the examples is the gastrointestinal tract. Another form of the single unit smooth muscle that is present in the uterus, that resembles the action potential in case of cardiac muscles. So whatever action potential I discussed in case of cardiac muscle, same type of action potential is seen in case of uterine smooth muscles, which is having a extra phase that is known as the P2 phase, that I am not going to discuss here. So next one is the action potential in multi-unit smooth muscles. Action potential in multi-unit smooth muscles. As I told, multi-unit smooth muscles are present multi-unit smooth muscles are present on the lining of the respiratory tract, iris muscles, piloerector muscles, which do not have any gap junctions, so they cannot act in concert they contract single-handedly, and this type of potential changes is not known as the action potential. That means in case of multi-unit smooth muscles, no action potential is developed. Rather, the potential difference that is seen, this is known as the junctional potential. So potential difference that is seen in case of multi-unit smooth muscle is known as the junctional potential, which is initiated by the neurotransmitter, that is nerve fiber. So for example, here you see this is the nerve fiber, axons of the nerve fiber, axon terminals, and this is suppose the smooth muscle. So at the action terminal of the smooth muscle, multi-unit smooth muscle, there is a release of the neurotransmitter. So these are the neurotransmitter. So once this one is stimulated, action potential will be developed there, and that action potential will pass to the synaptic terminal. That will cause the release of neurotransmitter at the junction between the neurons and the smooth muscle. Now this neurotransmitter will bind with the receptor present on the smooth muscle membrane. So on the smooth muscle membrane, there is a receptor. So there is a receptor present in the smooth muscle membrane, and this neurotransmitter gets bind with this neurotransmitter binds with this receptor. That means this is initiated by the neurotransmitter. So how it happens? Neurotransmitter release. This is followed by binding of neurotransmitter to the receptor present on the wall of the smooth muscle, and this binding of the neurotransmitter receptor complex, they is responsible for opening of ligand gated. Now here ligand gated calcium channels will open. Ligand gated calcium channels will open. So calcium entry will take place, and that is responsible for contractions of that particular multi-unit smooth muscle. So what's happened? These membrane potential at this membrane that is getting developed is induced by the neurotransmitters, and that type of potential difference is known as the junctional potential. So this is known as the junctional potential, and this junctional potential is initiated by the neurotransmitter release. So neurotransmitter once will be released, this will bind with the receptor. So this hormone receptor complex is responsible for opening of ligand gated calcium channels. So calcium entry take place, and that results the contractions of that particular multi-unit smooth muscles. Not only that, so this is number one. Secondly, what's happened? This neurotransmitter, once the neurotransmitter are released, they will activate. This will activate the G protein present on the present in the multi-unit smooth muscle membrane, multi-unit smooth muscle membrane. In all the membranes, you know, there are G proteins. Similarly, in the smooth muscle membrane also there will be G protein. So this neurotransmitter will will activate the G protein. Once the G protein will be activated, then what's happened? They will responsible for activation of membrane bound phospholipase C enzyme. Phospholipase C enzyme. So in the interior of the membrane, there is an enzyme remains in inactivated form, which is known as the phospholipase C. So this phospholipase C will be now activated by the activated G protein. Now what's happened? What does this PLC enzyme will do? This phospholipase C enzyme will cause the breakdown of the membrane phospholipids. On the membrane, there are different type of phospholipids. So some of the phospholipids will be broken down by this phospholipase C enzyme. So breakdown of phospholipids, membrane-bound phospholipids to inositol triphosphate IP3. Inositol triphosphate will be generated, will be synthesized, and this IP3 will result the release of calcium ion from sarcoplasmic reticulum present in the cell membrane. Within the cell there are sarcoplasmic reticulum. So this sarcoplasmic reticulum generally stores the calcium ion. So this calcium ion will be now released by the help of this IP3. So by the help of IP3, sarcoplasmic reticulum will now release the calcium ion, and this calcium ion is now responsible for contraction of multi-unit smooth muscle. So contraction of multi-unit smooth muscle is initiated by the release of the calcium ion from the sarcoplasmic reticulum. So by these two mechanisms, one is the calcium entry from the extracellular surface to the cell itself, inducing by the opening of ligand-gated calcium channels, and secondly, the neurotransmitter will directly responsible for release of the calcium ion from intracellular storage of the calcium ion. That is, storage site is the sarcoplasmic reticulum. So by these calcium ions, by sources from both the calcium ion, results the contractions of the smooth muscle, multi-unit smooth muscle present in the iris muscles or the linings of the respiratory tract. Now here one thing is also important, as I told, that calcium channel, opening of the ligand-gated calcium channels, they result the calcium entry. So calcium ion will enter from the extracellular surface, calcium ion will enter to the inside of the cells. So calcium ion will enter directly from the extracellular surface. Now this calcium ion will also help to trigger the calcium release from sarcoplasmic reticulum. So calcium ion from the extracellular source also triggers the release of the calcium ion from the sarcoplasmic reticulum, and that mechanism is known as the calcium-induced calcium release. So here this calcium entry is responsible for release of the calcium ion from sarcoplasmic reticulum. So this calcium entry, extracellular fluid, is responsible for again the release of calcium ion from sarcoplasmic reticulum, and this mechanism is known as calcium-induced calcium release. So this is the mechanism which is known as calcium-induced calcium release. So calcium ions are getting released in the intracellular part of the smooth muscle, multi-unit smooth muscle. How? from two sources. One is the intracellular source, another one is the extracellular source. Extracellular source also induce the release of calcium ion from intracellular source, and thereby by this mechanism enough amount of the calcium ions are getting released, and that calcium ion is ultimately results the contractions. So here contractions of the multi-unit smooth muscle are taking place, multi-unit smooth muscles are taking place without developing any action potential, rather there is a change of the membrane potential. Then that event is known as the junctional potential. So that event is known as junctional potential. I hope the concept is very clear to you regarding the multi-unit action potential in the multi-unit smooth muscles, or membrane potential changes in the multi-unit smooth muscle, and also the membrane potential changes takes place in the single unit smooth muscle. If this concept is very clear to you, you like this video, don't forget to subscribe this channel, and also don't forget to press the like button so that I can keep motivating to bring the next videos on different other topics. Thank you.